we're going to look for the population with the highest rate of the HIV virus.
That'd be nice on the show.
And it's really simple, actually.
So finding out where HIV is most prevalent. This is where AIDS is most likely. So random sampling, taking 1,000 people from random parts of the world.
You can check the percentage rate of the infection of HIV, and that's how we would see which area had the highest percentage rate of HIV, and that's where it's rated. And that's it.
What was your question? Wait.
It's just ridiculous. Nothing to do about it. Okay. Hi, I'm Linnea Wallace.
Fernando.
Patricia Gregory. Okay. We're gonna ask our question. Okay. Our question was, what is the chance that HIV infection will be able to be cured, and how? Okay. Oh, jeez, it's not like banana time right now. We'll have that time in a minute. Okay, this is an HIV virus here. This is RNA, as RNA here, and this is reverse transcriptase, which is also pictured there. Okay, okay. Our hypothesis is that it's okay. We don't think that it's likely that HIV will be cured because sorry. There's too many mutations and too many variables going on there. Fast rate of reproduction of HIV virus, mutation rate, okay, and many strains of the virus. So it's more difficult to find a cure that'll go for it won't work for everybody because there's different variables going on there.
So what we did, it was kind of hard to find an experiment that will prove decisively that it can be cured because it's really hard to do that. So we kind of did the opposite. We're going to try to do an experiment. We're trying to do things that could probably cure it and see the limitations of those approaches. So here are some things that we brainstormed and we came up with things that could be used to try to see if it could be cured. So first of all, like we already talked about Northern European people that are immune, that seem to be immune to the disease. We could use those as controls in our experiments and trying to find specific things that will block the CCR5 or the CXCR4, which is, I think, one of the mutated HIV viruses also gains access to the cell through that protein rather than the more common CCR5. And when we're talking about it, this is a plot of the HIV virus, and there's a plot of the T cells. Yeah. So this is the T cell, the HIV virus, and we were thinking if we had any chance of curing it at all, this would be the time of that, because the T cells are relatively high, and yeah, and the HIV is low here. So here, this is the place. So this is like the place that we would try to do it, but we didn't know how early in the process that was, and so we would have to have really good testing techniques and stuff like that. So this, if we thought if there could be a cure, this would be the time to do it because we could help our immune system itself fight it off, and that was our thing about catching it early. And this is just like pills that they use to that currently, like the treatments do some of the things. So they block the entrance to the cell. They also block reverse transcriptase, and they block when the DNA from the virus gets into the genome. So these are just the pills, and maybe if those can be improved, then those could help here and maybe widen the gap where our T cells are higher, our HIV is lower, and hopefully that's the only place where we could see that actually work. But again, we thought that chances of that were really slim.
Okay, so as far as like, excuse me, so in regard to conclusions, just like Fernando said, within finding a cure, other factors have to be involved, such as we need, well, the system for testing HIV is efficient as it is now, but in order to catch it early, HIV tests needs to be reorganized so you will be able to test HIV within a week, because you know they say get tested for HIV between every 3 to 6 months, because once you get infected, it doesn't appear until 6 months later. But by the time it appears in your body, it's too late, and that's when the mutations and everything else. So along with better testing techniques, you also have to do a lot of research, because different people from different geographic areas have different, not DNA makeup, well yes, well I guess different DNA makeup, and along with different strains and things like that, because the simple fact of how the virus is so smart. The only way to catch a cure is if you catch it early, I guess during the incubation period. But that goes back, you can't catch it during the incubation period, because during the incubation period, you don't know you have it.
Questions? On the graph of where you would most likely cure the disease,